Still on the lectures on the renal system. Today we'll be dealing with the structure of the nephron. The structure of the nephron. Alright, so let's look at what the nephron is all about. So basically the nephron is the structural and functional unit of the kidney. And we have about 1 million nephrons in each kidney. 1 million. So putting both together, we have about 2 million nephrons in our kidney. So the nephron is made up of two basic things. We have the renal corpuscles, which is made up of the glomerular capillaries and the Bowman's capsule. Then we have the renal tubule, which is made up of the proximal convoluted tubule, the loop of Henle, distal convoluted tubule, and the collecting duct. So this is just the basic structure of the nephron. The basic structure of the nephron. Now we have mainly two types of nephrons. We have the cortical nephron found in the cortex. Then we have the juxtamedullary nephron found at the juxtamedullary junction between the cortex and the medullary. But other books will add another type called the subcapsular nephron. But basically it's two. And we have 85% of our nephron being in the cortex and 15 or 10% being the juxta, and then the remaining 5, if it is there, being the subcapsular nephrons. Subcapsular nephrons. So basically that is what it is all about. So now let's dive a little bit deeper into the renal corpuscles. So this is just a typical structure to illustrate to us what the renal corpuscle is made up of. So as we said earlier on, the renal corpuscle is made up of the glomerular capillaries or the glomerulus, and then the Bowman's capsule. The Bowman's capsule. So the glomerulus or the capillary system is made up of fenestrated endothelial cells, the basement membrane, and in between them we have the mesangial cells. The mesangial cells. So they can be found rarely in the middle, called the intra, and then outside called the extra mesangial cells. The extra mesangial cells. Alright, so let's continue to go deep. So let's talk about the basement membrane or the glomerular basement membrane. It is composed mainly of 3 layers. 3. We have the lamina rara internal, which is just adjacent to the endothelial cells, contains heparin sulfate, and this makes it a negatively charged molecule. Then we have the middle layer. Unfortunately, they are not shown over here in this diagram. The middle layer consists of type 4 collagen fibers and laminin. They prevent the passage of large protein molecules. Then we have the lamina rara external, which are adjacent to the podocyte processes, also contain heparin sulfate. So together it makes this membrane a negatively charged membrane. Negatively charged. So this prevents substances that are negatively charged to actually pass through, because they will repel them. They will repel those substances from entering. So usually positively charged molecules can easily penetrate through this membrane. Then let me just add these ones to it. So right above it, you can see the distal convoluted tubule, also made up of what? The macula densa. It's not part of the renal corpuscles, but at the long run you will see them as well. And this macula densa is able to sense sodium ion concentrations. And we have the juxtaglomerular cells, also helps to regulate the vessels. Alright. So still on it, let's now talk about the messenger cells. These messenger cells have what we call the contractile cells, which helps to regulate blood flow and secrete extracellular matrix, prostaglandins, and cytokines. They also have phagocytic activities, which helps to remove proteins and other molecules that are trapped in the glomerular basement membrane or the filtration barrier. The filtration barrier. The Bowman's capsule. It has two layers. We have the outer parietal layer, then we have the inner visceral layer. The inner visceral layer is composed of a modified squamous epithelium known as podocyte. They are just attached to the membrane or the glomerular membrane, immediately attached to it. Now, these podocytes have what we call foot processes, which interdigitate with other podocytes or adjacent podocytes to form what we call the filtration slit. These allow even much smaller molecules to pass through, because the fenestrated capillaries has a larger fenestration or a larger hole than the filtration slit. And in between them is what we have the Bowman's space, where the filtered molecules actually pass through, then it enters into the proximal convoluted tubule, and then is excreted as urine. So let's take a look at the filtration barrier, because having a firm understanding of it also helps in disease processes. So this is just the capillary system, and then this is our barrier. So what do we have here? We have the fenestrated endothelium of the glomerular capillaries, which is, of course, this is about 70 to 100 nanometer, if I'm right, 70 to 100. So we have the fused basal lamina of endothelial cells and podocyte. Then we have the filtration slit. Now, in between them, we have this diaphragm. So the whole year at the filtration slit. Then in between them, we have what we call the slit diaphragm, or the diaphragm, which further further filters the molecules that pass through them. So now this barrier permits passage of water, ions, and small molecules from the bloodstream into the Bowman's space. And of course, large and or negatively charged proteins are prevented from passing into the Bowman's space. Why? Because proteins are negatively charged, and secondly, they are large in sizes. So definitely they can't be allowed to pass through. So viewers, basically this brings us to the end of the nephron, or the structure of the nephron. And in next lecture, we will talk about the renal tubule in details, where we have the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Guys, you want to know more about the renal tubule, so please do well to follow us on our social media platforms, on Facebook at Ro1, on Instagram at Ro1_Ro1, on Twitter at RowanOurWorld1, and on YouTube at Ro1 TV. For YouTube, do turn on the notification bell, because every now and then we post videos and you don't want to miss them out. So please turn it on and be the first to view. And if you like it, let us know. If you have any question, let us know. If you want us to improve on some things, please do let us know. We are doing this because of you. All right. Thank you.